All right, so we will go ahead and get started. Sorry, everyone, we have had some issues connecting to our Facebook Live in the past couple of days. So we are just going to host this on Zoom, and it will be available to watch again after the meeting is complete. I'll post it. Please send in questions, and then Dr. Cooke, I will, if it's okay, send you, if we have any additional questions that come in after it's posted, I'll just send them your way to get them answered.
That's fine.
Perfect. So hello, everyone, and welcome back. Today we have with us on Ask a Scientist, Dr. Cooke. She is a professor of functional anatomy and works on her research with primate evolution in the Caribbean. And she is, I'm very excited to have her as a guest today. So I'm just going to go ahead and pass it off to you, and I will monitor the chat and just let you know.
Great. Hello, everyone. It is nice to be with you this morning. I'm going to start my screen share here so you can see what I have. All right, here we go. Share, and then I will start my view. Play from start. And all right, everybody see it okay? Excellent. Okay, so thank you for being here with me this morning. I'm coming from Baltimore in my house, in my basement. I will start to talk to you about the primates of the Caribbean and my understanding their diversity, past and present, and their eventual extinction. And while my research is primarily focused on primates and has been for quite a few years, I am starting to do some work on other animals as well from the Caribbean, including sloths and rodents, so I'll talk a little bit about them. So here's our plan of attack. I'm going to start out talking about the evolution of the Caribbean basin, both from a geological perspective and from the perspective of the native mammals. Then a little bit about the Caribbean primates, and then understanding ecological niche and some of the methods that paleontologists and biological anthropologists use to do that. And then finally, we'll finish up with Caribbean extinction and talk about some of the things that may have resulted in the extinction of the Caribbean mammals. So first off here then, the evolution of the Caribbean basin. So this is the Caribbean, and I've taken this image from the Google Maps, and we have up here southern North America and northern South America, and then the Central American land bridge that connects North and South America here. And then this is the Caribbean basin. And in this image, you can actually see the outline of the Caribbean plate, the tectonic plate here. And along the leading edge of it, there are a number of islands and island systems. And these include the Greater Antilles, which my talk is going to concentrate on today, as well as the Lesser Antilles outlined there in yellow, which is a series of smaller islands. And then we also have the Bahamas and Turks and Caicos, which are shown in red there. And all of these island systems have native mammals on them, including rodents are the most numerous, and so are bats. Well, I'll go back one. This is what the Caribbean looks like today, but it didn't always look like that. And this is an approximation of what the Caribbean islands would have looked like about 31 to 33 million years ago. And you can see that North and South America at this point are separate continents that are not connected to each other. And they have endemic fauna that is present just in North America and just in South America. And I'm going to be using the term endemic a lot throughout this talk, and that means that that animal is just found in that region. And so we have this separation. And between North and South America, the Caribbean plate, which would have been right in this area here, is starting to slide through between them. And again, along the leading edge of it, you have land masses. At this point in time, there may have been a connection, either a solid land connection or a series of very closely spaced islands that connected the Greater Antilles which includes Hispaniola, Cuba, Puerto Rico, and parts of Jamaica, to South America. And here, this is taken from a reconstruction done in a paper by Manuel Israel de Vinet and Ross McPhee, and I've colored it here so you can see the land and the sea. This possible land bridge or possible group of closely spaced islands was present just for this short period of time. By about 11 million years ago, the land bridge had subsided, and there was just a small portion of the Greater Antilles that was subaerial or above the water. Land masses were smaller during this period of time because sea levels were higher, and so a lot of the land was inundated with the sea. You can also see at this point where you start to get a few islands here in what is today Panama that would eventually come above the water to form the land bridge between North and South America. Now, the Greater Antillean fauna includes, or the mammalian fauna at least, includes a number of animals that are usually, with the exception of Selenodon, are derived from the South American mammalian groups. So we have primates, ground sloths were present on the islands. There were some small insectivores or insect-eating animals like Selenodon here, and there were a number of now extinct shrews called Nesophantes. A good number of rodents here. This is a living one, Plagiodontia diem, which is from Hispaniola. It's found in, on that island. And then lots of bats as well. And this is it for the mammalian fauna on these islands. And today, about 72% of the endemic mammals are extinct, and about 85% of the non-volant or non-flying mammals. Overall, the Greater Antillean fauna is depauperate, meaning that it's not particularly diverse compared to the mainland populations and fauna. So it lacks a couple major mammalian groups, including carnivorans, which are carnivores, things like cats and dogs or wolves, mustelids, which are small animals like weasels. Those are the carnivorans. Nodoungulates, which are hoofed mammals that were endemic to South America. Marsupials, so animals like possums. And then cingulates is a large group that includes armadillos, and they were very numerous in South America as well. So none of these animals are found in the Greater Antillean fauna. But the animals that we do have there are largely South American in origin. And this is an image, a reconstruction of Megalonychus rodens, which is a large ground sloth. Would have been about the size of a large bear. So one of the major questions in understanding the species in the Caribbean revolves around its biogeography. And biogeography is a study of the distribution of species across space and through time. And many researchers have been interested in how all these Antillean mammals arrived on the island. So how did they get there? And then once they were there, how did they adapt to these new island environments? And then second, how are they related to the mainland forms? There are a couple of different hypotheses for how the animals arrived in the Antillean islands. One option would be something we call vicariance. And this hypothesis suggests that the animals would have walked across this land bridge, the Garlandia land bridge, which I showed you in that previous slide, that was present about 31 to 33 million years ago. And vicariance is a biogeographical term which refers to how animals move from one region to another across something like a land bridge, and then are separated by some sort of geographic barrier at a later date. So it divides those two populations. The other option would be something called dispersal. And dispersal generally occurs across water. So these animals disperse or move across a pre-existing geographic barrier. And that would be an option too. So if we were to look for evidence for vicariance versus dispersal in these populations of mammals, we'd have several expectations. One would be for dispersal, we would expect that the population of animals would be depauperate. It wouldn't fully reflect the mainland fauna. And this would be because not all mainland fauna would be capable of dispersing across this geographic barrier. So some animals, like bats, would be able to fly across it, and we would expect them to disperse easily. Animals like sloths have been known to be good swimmers. And so potentially they could, at least for a short period of time, survive an overwater crossing. Rodents are small enough, for the most part, that they can disperse across a open water boundary by potentially rafting, or if you can imagine a big storm where trees and bushes and clumps of roots and things like that get blown off a mainland, a few small mammals might get blown with them. And so they could survive on this raft of vegetation, and if they were lucky, land in an island environment. So this depauperate fauna would be a fauna that were capable of dispersal. We would also expect to see different evolutionary divergence states between the mainland forms and the island forms. So you can use something called a molecular clock when you are comparing two different strands of DNA from different populations of animals or different species. And based on the number of mutations that you see, you can back calculate to figure out how long those populations or those species have been separated from each other. Because genetic mutations in DNA happen at approximately an even rate. So if you see, if you know that there is one mutation every 2,000 years, I'm just making numbers up here, and you have a certain number of mutations there, you can just use a mathematical formula to calculate how long those populations have been separate from each other. So we would expect to see lots of different evolutionary divergence states. Now, if vicariance were the best explanation, we might expect to see a continental fauna. So there was no geographic barrier, so basically anybody could just walk right over to that new area of land. We might also expect to see a single series of divergent states from the DNA, because that new area of land was colonized by these different populations all at the same time. So we would see a clustering of divergent states around the period of time where this colonization occurred. Because the timing for this potential vicariant event would be clustered around 31 to 33 million years ago when the Garlandia land bridge was present, we would also expect to see that the fauna on the Caribbean islands looked more similar to a fauna on the mainland that was dated to that period of time. So we would expect potentially that this Caribbean fauna could have some primitive characteristics that were maintained on these island environments. So far, let's see what we've found. The evidence from rodents largely supports a dispersal. And a lot of this work was done by a colleague, Nate Upham, and some of his colleagues as well, like Bruce Patterson. They published I just realized I have a typo. It's not Patterson, it's Patterson. I'll fix that later, sorry. In 2015, they published a nice phylogeny of South American rodents and found that many of the Caribbean rodents seem to have diverged from the mainland around 17 million years ago or so. So that's quite a bit after this land bridge would have been beneath the waves. And these are two modern species that are still alive in the Caribbean. We have Plagiodontia here. This is found in Hispaniola. And then Geocoprome is brown eye, which is found in Jamaica. The evidence for bats is more complicated. Liliana Davalos has done a lot of work on the bats, looking at genetic divergent states. And her work supports both dispersal at a later date in the Miocene, that period of time that encompasses about 23 million years ago to more recent times, and as well as some very ancient divergences. So it's possible that the bats may have traveled up that land bridge about 30 million years ago, and also other populations came later. This is not terribly unexpected for an animal like a bat, which would be good at dispersing over water boundaries. It may have been blown off course, or may have kind of island hopped along the Lesser Antilles. Yes, I see I'm being waved at.
We do have one question. We have Molly asking, is a land bridge the only way that the vicariance could occur? Sorry if I missed this.
No. Yes, more or less. In the Caribbean basin, that is what we would be talking about. You can have vicariance. The term just refers to a population that is divided at a later time by a geographic barrier. So in the Caribbean, the geographic barrier would be water. In other areas, it could be something else, like a desert or a mountain range or something like that. But generally speaking, when we're talking about vicariant events, we normally are referring to, in the Caribbean, water boundaries. Any other questions, or we're good?
I have one myself. Can you
sure.
briefly describe the difference between the vicariance and like something like allopatric speciation?
Oh, sure. The vicariant event is really referring to a vicariant event would result in allopatric speciation, essentially, where the vicariant event is really referring to the biogeographical context in which speciation would occur, if that makes sense. So allopatric speciation is a type of speciation, just for the other folks, where a population is divided and then they can no longer exchange genes by mating with each other, and so they might go in different directions over time genetically. Or might adapt to local areas, and there's lots of things that can cause that. But that's more or less how they relate. Okay. Now the evidence for primates is more limited, and I'm going to talk in detail about the primates. But the primates are really weird. They are very different from the primates that we see on the mainland. And so far, the only way of looking at their genetic related lists would be to find a fossil, or a subfossil really, that still has some DNA in it. And in tropical regions, DNA will degrade in hot, wet environments. So many of the caves in which these fossils are deposited are hot and wet, and so the DNA degrades and you can't pull it out of these fossil specimens. But there is a species called Xenothrix mcgregori, which was found on Jamaica, which is it's relatively young. It went extinct. The last date that we have for it is about 1,400 years ago, closer to 1,500 actually. So those fossils, or subfossils as they are much younger. And researchers, Woods et al., recently published a study where they looked at the ancient DNA from this fossil Xenothrix and compared it to modern South American primates. And they found something really interesting and unexpected. Researchers have been arguing about whom Xenothrix was related to for a very long time. And lots of folks thought that they might be related to this group Callicebus. But what Woods et al. Found was that they were nested within this great big phylogeny of this very diverse group of monkeys, the TD monkeys, genus Callicebus. And so they were more closely related to some Callicebus than to others, because they're within this whole tree of Callicebus. But the thing that makes this interesting, you might be like, okay, fine, it's related to some South American monkeys, big surprise. But the thing that makes it interesting is that Xenothrix is very different morphologically from the Callicebus on the mainland. And these Callicebus monkeys are, all fairly similar to each other. They're about a kilogram or so. They have 3 molars. They have a very particular postcranial morphology where they're not particularly robust or heavily built. And Xenothrix is none of these things. It's about twice the size. It has a very different dentition where it's missing some molar teeth, and has a really heavily built skeleton. So what this tells us is that when this ancestor of Xenothrix got to Jamaica sometime around 10 million years ago or a little before, it adapted and changed a lot to its new island environment. It got much larger, which is a phenomenon that we see in a number of island species. And it lost some teeth, and the morphology or the anatomy of the teeth changed. So this evidence supports dispersal. Now, to go a little bit into more detail about the primates, we have Xenothrix. Let me get my mouse going here. There it is. Xenothrix is down here, and it's found in a number of different spots on Jamaica. These little black dots are fossil sites where we found different primates. On Cuba, there's a species called Paralawata veroni, which is shown by this skull here. And then there's also another species, Paralawata marianae, but that one is only known from a single ankle bone, so I can't say as much about its adaptations. And then on Hispaniola, there is Antillethrix florenensis and Insulacebus tusantiana. Now, one of the interesting things about these primates is that we think that they went extinct relatively recently. There's a good radiocarbon date for Xenothrix, which places it alive on the island 1,500 years ago. And that is when, about the same time that Amerindian populations, or the native people of Jamaica, started to colonize that island from South America. And it is possible that some of the primates survived on the islands until the arrival of Europeans. So we have a manuscript, it's late 16th century, the Drake manuscript, which has some records of primates. And one of the really interesting ones that I like is this. They write, this is in French here, and they write, "This is a very agile beast. The Indians cannot tame it or feed it, for they die of grief and do not eat in captivity. They have a face like a human and are very mischievous and rebellious and throw themselves at people, mainly against their faces and eyes." So if the primates arrived or survived until the arrival of Europeans, and not too much long after, it seems like they put up a good fight on their path to extinction. But we do have this evidence, and it's an interesting illustration, not entirely accurate. You can see it has teats or mammary glands. It has 4 of them. We know primates only have 2, but it does have a human-like face here, and or a primate-like face and a long fluffy tail. So it's some interesting historical evidence. Now, I mentioned all of these primates. So we have Paralawata veroni and marianae, and this is an interesting primate. It would have been about 5 to 10 kilograms, so pretty big. And it shows some shape similarities in terms of its cranium to the modern howler monkey. So some researchers have suggested that it is a howler monkey that got to the islands potentially by dispersal fairly early on in howler monkey evolution. And some of the similarities you can see is it has a pretty small brain case, like a howler monkey. This over here is a modern howler monkey. It has this, what we call aerorhynchus, or kind of scoop-shaped face here and there. It has some other shape similarities, which I won't go into detail, in the base of the cranium as well. But that's where the similarities between Paralawata and modern howler monkeys end. Postcranially, or its skeletal anatomy basically below the neck, its arms, its legs, and its hands, are fairly unique compared to mainland primates. Platyrrhines is the technical term for South American primates. And it has this really heavily built skeleton, kind of similar to Xenothrix, and it has really short fingers and toes, which some researchers have suggested means that it might have been terrestrial or ground dwelling, which is also a unique way of moving around for a South American primate. Dentally, its teeth indicate that it ate fruit, or it was a frugivore, but it may have had some dietary flexibility. And that's in contrast to what we see with the living howler monkeys, which largely eat leaves and have very unique dentition adapted to leaf eating. So there's some similarities with a modern form, but we don't really know if that is a result of a shared evolutionary history, or if it's what we call parallelism, where they developed these characters in parallel with each other because they had similar adaptations. Now here, yep. Go back to our parallelata.
Yeah, so we had a methods question come in from Molly. And so it says, now that DNA is used so widely, is the functional morphology field more dependent on DNA, or is the emphasis still on shared physical characteristics?
It depends. For fossils, sometimes we can get DNA and sometimes you can't. And so for the fossils that are either too old, so DNA is not preserved, or just the preservation is not good. They might be young fossils, but the preservation just isn't good. We still have to understand evolutionary relationships. We still end up using morphological characteristics. Now, in terms of using morphology to understand things like locomotor behavior, we generally still look at how that anatomy helps that animal move around in their environment. Now, there certainly are some researchers who are working on gene expression and those kind of things to better understand how animals and humans move in their environment, and even potentially things like muscle function and so forth, as well as the development of the postcranial anatomy genetically. But that field is actually still fairly young, so it hasn't widely been applied to understanding fossils yet. Okay. So I'll move on to my next one. We know about Xenothrix already. I already mentioned him, but I'll tell you a little bit more about its characteristics. So it has this amalgam of primitive and derived characteristics. So primitive things are ancestral characteristics, and derived characteristics are those that are unique to that particular species. And this is the lower face and palate that I showed you before. So it's just this portion of the anatomy down from, this is the nose and the front teeth and the cheekbones, here the zygomatics, and then some molar teeth. And this is a view looking down, or looking sort of from an inferior view from underneath the palate. And so we have some premolar teeth and some molar teeth. And so you can see that it has just 2 molars. It has a decreased dentition. And this species, as I mentioned earlier, is postcranially robust. And its teeth are pretty interesting. They're very wide and flat, and are very unlike any other living species. There isn't another primate species that has teeth exactly like this. And so we think that it probably ate some hard objects, something that it could crush with its dentition, because it has these wide, flat teeth, but we don't know for sure. And then finally, primates which I have spent a little more time studying include Xenothrix bernensis and Insulacivus tusantiana. And these are both from Hispaniola, which is the island that has the Dominican Republic and Haiti on it. And this is a skull, the first skull that was recovered of Antillephrix bernensis. And the postcranial morphology of this species indicates that it's kind of a generalized arboreal primate. There's nothing that was particularly special about its postcranial morphology that would let us say that it was uniquely adapted to, say, leaping, for example, or dwelling on the ground. It's kind of a run-of-the-mill arboreal or tree-dwelling primate. And then dentally, its teeth, both species, look quite a lot like some of the South American primates from the Miocene, or from about 15 million years ago or so. So there isn't a lot about these particular species that allow us to tie them into either a living group or a fossil group. Now, I have spent quite a lot of time working on Hispaniola, and that's where this guy comes from. And this is our island of Hispaniola. The Dominican Republic is over on the eastern side, and Haiti is on the western portion of it. And the boundary line is well, sort of like that. At one point, it was two paleo islands. There was a northern island and a southern island. And that was divided by the Enriquillo Graben, which is a depression. And actually, if we look back, you can see even today that this area, it has some lakes in it, so it's lower elevation, but it also, you can see that there's less tree covers here. It's a little bit browner in this map, whereas just north of it, it's nice and green, and south of it, it's nice and green. And part of the reason for this is this soil here, because it has been inundated with the sea periodically, is much saltier, so less, fewer plants grow in this area, and those plants that do grow there have to be adapted to that salty soil. So we have our two paleo islands, Enriquillo Graben, and then these are where the primate fossils are found on Hispaniola. And a number of researchers have worked here. The little yellow stars indicate sites that my colleagues and I have worked at for the last 11 years or so. And in the 1980s, there were researchers from the University of Florida, Gainesville, that did a lot of work on the, this is called the Tiburon Peninsula, the southern peninsula in Haiti along there, and collected many, many fossils, including the specimens which I eventually named as Insulacebus dessantiana. In the 1970s, out of the Museo del Hombre Dominicano, Renato Rimoli and colleagues worked at a cave here, Cueva de Berna, and this is the site that the first fossils of the cranium of Antillothrix were found. And then in the 1920s, there was a researcher, Garrett Miller from the Smithsonian, who collected quite a few specimens. We don't know exactly where all of his sites are, because he describes where the sites are, but doesn't, of course, this predated GPS by quite a lot. So he doesn't always give map coordinates or anything like that. So researchers don't exactly know where all of his sites are, but he had a number in the central region here of Haiti, and then some along this peninsula in northern Dominican Republic. So my field work takes place in caves. Most of these fossils are recovered from underwater caves, although my colleagues and I also work in dry caves. And these are caves from the easternmost Dominican Republic in Alta Gracia province. This cave site, this picture turned out kind of nice, and the cave divers were underneath the water here, and so you can see the lights that they have illuminating the water. And this is what an entrance to one of the sinkholes might look like. They all have different types of entrances. Sometimes it just looks like a little lake, but it goes down. And this site here, actually, you had to climb down into the hole, and this is a little ladder to get down in. And then the cave opens up once you're in. So I collaborate with a number of cave divers, Philip Lehman, Christian Pitaro, Zach Kluckert, who do a lot of the cave diving. Here are some other images from a site called OLEG'S Bat Cave. And Oleg was one of Philip and Christian's collaborators. And this site, it had many, many bat remains. And here, this is an image showing the bats. And so you can see these little dark spots here are all of the skulls. And then these are long bones, a lot of wing bones, long phalanges or finger bones that make up the wings for bats, and some femora and things like that. and here. There are many, many bats. There were also crocodiles found in this cave. So here's a bit of a croc rostrum sticking out of the mud. And then I'm not sure if this is, I think this might be Philip, but I'm not 100% sure. I can tell that's their logo for the Dominican Republic Speleological Society.
I feel like that's the jackpot cave.
Yes, many of these caves are extremely rich. And part of the reason they are rich is because as these animals filter down to the cave floor, they're protected essentially by the water because when the researchers go in to collect things, they can just float above the cave floor like here. And then there's no risk of harming these fossil specimens. But yeah, many of the Dominican caves, both the wet and the dry, are very, very rich with fossils. And part of that is also that there's sinkholes. So there's a hole in the ground, and then these animals will come and walk along and fall in and become trapped. And after, thousands of years, lots and lots of specimens accumulate. So Antillethrix bernensis for many years was known from just a single specimen of a part of a maxilla or the upper jaw. But starting in 2009, my colleagues and I, and then another team as well, started to explore these caves that had water, and a number of skulls have been recovered. This is the first one that was recovered. It's actually a juvenile, so you can see the sutures or the space between the bones and the skull are still quite open, indicating they could have grown a little bit more. After this, we were also fortunate enough to find a tibia or a lower leg bone that was encrusted in a speleotheme or rock that the cave forms. And many of these speleothemes can be dated using radiometric dating techniques. And so we collaborated with Robin Pickering, and Helen Green, who are both specialists in dating different types of cave deposits. And they found that this tibia that was encrusted in the speleotheme was about 1.3 million years old. So some of these specimens are actually really quite old, but others are much younger. So it seems that Antillethrix was on this island for quite some time. Most recently, we found a number of new crania, and my colleague Lauren Hallinar and Zach Pluckert and I have been working on some of those. And let's see, so here's a new skull. So this one you can see is much more complete. One of the things that you can note about it is it has a relatively small brain case compared to many of the mainland forms. This is another palette we have of one of the skulls. And then this specimen right here is the type specimen, or the specimen that was used to describe the species that Renato Rimoli found in Cueva de Berna in 1977. So for many years, this was all that was known of this primate, just this little specimen here. But now there's about 6 full skulls and many, many postcranial elements. As part of this project, we commissioned a reconstruction about what this might have looked like. We certainly can't know what its fur would have been like or anything like that. But based on the postcrania, it might have been something similar to a Callicebus or a TT monkey. So David Reaney, who is a professor in the Art is Applied to Medicine department at Hopkins, did a reconstruction of what it might have looked like. Now, on the other side of the island, we have Insulacebus tusantiana, which is much more poorly known than Antillethrix. We have many fewer specimens of this. There's a femur, or an upper leg bone, and a nice dentition shown here. Everything that's dark gray is a tooth we have, but then in making this figure, I mirrored it so I could make a full tooth row. So the light gray is just a mirror of the other side. And these specimens have some small differences from Antillothrix. Overall, they're about the same body size. They would have clocked in around 3 to 5 kilograms or so. But it has some differences in muscle attachment area on the postcrania, on the femur, and then on the humerus, which is the upper arm bone. And then there's some subtle differences that I won't go into in detail in the dentition as well. What's interesting about this is this follows biogeographic patterns of animals across the island of Hispaniola. All of the specimens for Antillothrix are found mostly on the eastern part or the northern Paraleo island, whereas the specimens for Insulacebus are found in the western southern Paleo island. So they might have been separated, these different populations. I do think they are probably very closely related to each other. Now finally, I'll finish up talking a little bit about ecological niche. So here we have some different Calicebus or TT monkeys demonstrating different aspects of ecological niche. Now there's lots of different ways of defining what ecological niche is mathematically. So ecologists will use different methods for modeling how an animal might sort of take up space in its environment. But sort of the most general way of thinking about this is that ecological niche is the role and position a species has in its environment. So this includes things like how they get their food and what they eat, how they reproduce, what their social systems are, how they move in their environment. In something like a South American primate that lives in a rainforest, like the TD monkey, where they live in the canopy can also be an important part of their ecological niche. Are they way at the top of the canopy, many hundreds of feet off the ground, or are they low in the understory where it's much more dense? So these are just showing you some different aspects of ecological niche here. For an animal that is extinct, it is more difficult to reconstruct ecological niche in some ways because you cannot observe what they are doing. You can't watch them mate or watch what food they eat. So we have to use a couple of different techniques to be able to try to reconstruct some of how they interacted in their environment. One of the things that you can do is look at dentition. And dentition our teeth are really informative about what an animal eats. And they also preserve really well in the fossil record. So we have lots of teeth of Paraloata, for example, but we only have one skull. And the reason teeth preserve really well is because they're hard. They're made out of a very dense enamel. And just as an illustration here, I've used two living primates. This is a howler monkey. You mentioned that guy before, and they're a leaf eater. And then this is a fruit-eating spider monkey. They are about the same body size, so they both are around 10 kilograms, 5 to 10 kilograms or so. And these teeth are actually at the same scale. So the first thing you might notice is that the howler monkey, which eats leaves, has much larger teeth than the spider monkey. And this is a lower molar, so one of these lower cheek teeth. And this is the occlusal surface or the biting surface, and this is the side of the tooth that faces the cheek. So you can see that it's much bigger. And then it also has really tall, sharp cusps with these long shearing crests along the edges. And here, and over along here. And then in the fruit-eating primate, you have shorter shearing crests, the tooth is smaller, there's less grinding surface area, and the cusps tend to be more rounded than in a leaf-eating primate. So using modern primates where we know what they eat and we know what their morphology is and we can get a good sample size, we can look at their adaptations and compare those to what we see in the fossil record to get a good idea of what something might have eaten in the past. So this diagram is a little bit technical, but one of the methods that I use for understanding teeth is to look at how tall the cusps are and how much surface area there is on a tooth. And I do this by taking lots of measurements across the surface of a tooth and then using a type of statistical analysis called principal components. Now, the principal components analysis will separate different morphological forms based on these points that I take across the tooth into different categories, essentially. Well, I tell it, what the categories are, but what I have found is that for one of the principal component scores, it correlates really well with the type of diet that an animal eats. So these colored bars across the top here represent different species of modern primates. And then what they ate is shown in these large colored areas. So pink is the principal component scores that fall in this area, correlate with a diet of hard objects, so things like nuts or really hard fruit. The blue region here correlates with principal component scores that indicate leaves or things like insects that are structural carbohydrates. So things that require a lot of mastication or chewing to break up. And then fruit-eating primates kind of fall in the middle here in this yellow. So there are areas of overlap, certainly, in the dental morphology between fruit-eating primates and leaf-eating primates. But there are not areas of overlap for animals that are really well adapted for eating structural carbohydrates, or hard objects. We can look at the distribution of those primates across that area. Now, if we look at where Greater Antilles primates fall, fossil primates from Patagonia, or fossil primates from a site called La Venta, which was about 11 to 13 million years ago and is found in Colombia, in a tropical rainforest, we can look at the community of these primates and see a different distribution of dietary types. So with the tropical forest extinct primates, we have a lot of spread. So there are some that are specialists at this end in leaves and structural carbohydrates. There are some that fall in this area of overlap, some that fall in the fruit eating area and into the area for hard object eating. So we have all of the different dietary types represented. In the Greater Antilles, it's a much smaller distribution of morphological forms. Mostly, these animals are all falling in this area of overlap between fruit eaters and leaf eaters, indicating that they have well enough developed cusps that they potentially could have consumed items that were more difficult to process. That's the same area of overlap that we find with extinct Patagonian primates, which were also potentially in a marginal environment or an environment that was not really very, very rich as a tropical forest would have been. Now, oh, yeah, there's a question.
So are the distributions based on like food availability? Is that kind of how that works, or like, yeah, okay. So.
Yeah, it would have been both food availability and species richness and diversity. So in a tropical rainforest, you have more ecological niches available, essentially, because you have a lot of fruiting trees. You don't have a lot of seasonality. There might be a wet and a dry season, but trees have leaves all year long. And so you tend to see a much higher degree of species richness and consequently more specific ecological niches, because that animal can be a specialist in that very rich environment. Whereas if you live in an environment that is more marginal, that has a high degree of seasonality, you can't as easily be a specialist because your food might not be available all year round. So if you're a fruit specialist, what happens when there's no fruit during the winter, right? So within animals that live in the Greater Antilles and Patagonia, those are much, much more northern or southern environments, and there would be a greater degree of seasonality. Coming from North America, we think of the Antilles as being, very lush and tropical, but not all areas of them are. And there is some degree of seasonality.
And then we did have a few questions just about the cave, if we can backtrack just a little bit. So we have
Sure.
one from Molly that says, do all the fossils come from drowning, or did they die in the cave before it was filled with water?
Probably they died in the cave before it was filled with water, but we can't know 100%. We know that these caves, many of them were dry at one point because they have cave formations that only, like stalactites and stalagmites, that only occur when the cave is dry.
And then we have one from Kinsey that says, are the caves both salt and fresh water? And is there a preservation difference?
Caves in the Dominican Republic that I work at are all freshwater. But there are caves in the Bahamas that are salt and fresh. And the saltwater caves in the Bahamas do have pretty nice preservation as well. I don't think anybody's done a good study to actually compare the difference between them. Good to go? Okay. Now, another way of looking at a diet and niche is using something called stable isotope analysis. And this is a project that I've been doing in collaboration with my colleague Brooke Crawley at the University of Cincinnati, and she's a stable isotope specialist. So we looked at the dental morphology of a number of Hispaniolan, specifically Haitian primates. And stable isotopes are different forms of an element, and they will get incorporated into the dentition or into the enamel of the teeth as the enamel is forming. And a couple of different stable isotopes will occur in different proportions depending upon what that animal ate. So we look at carbon, nitrogen, and oxygen. Another thing that can influence the stable isotope proportions are climate. So it can give you a lot of information about what that extinct animal ate and what kind of environment it was living in. So we have done some preliminary work using stable isotopes, but we actually just submitted a grant to expand this work this week, and hopefully that will get funded, and maybe in a couple of years I can tell you more about Haitian rodents. We'll find out if the NSF likes it. But our preliminary work included a sample of about 34 endemic species and one invasive, the rat, the Rattus norveticus, which is the Norwegian rat, and that was introduced when Europeans arrived on the island. And we sampled from 2 different localities, which are shown here, Truing Jeremy number 5 and Trou Jean Paul. And the terms truing and trou are Creole words that mean like a sinkhole type cave for truing, and trou is like a cave that you kind of walk in. Now, these endemic rodents that we sampled have a number of different body sizes. So they include some really large ones like Plagiodontia belozi, which is about between 5 and 6 kilograms. So that's a sizable rodent. And then some very small ones like Britomes, which is less than a kilogram. Oh, yep. We were able to get radiocarbon dates on a lot of these. So the rat, of course, the radiocarbon dates were 350 to 500 years ago. And then these other species, we were also able to get some radiocarbon dates, and they range from about 1,600 years ago to about 11,000. So the stable isotopes that Brooke isolated were carbon and oxygen. And these correlate, as I mentioned, with diet, so different types of plants, and different proportions of leaves and fruit versus fauna, and whether they might have been arboreal or terrestrial, because we get different proportions of oxygen isotopes if they're up in the canopy versus down on the ground, and whether the environment was drier or wetter. So what we found was interesting for a number of reasons. First, the rat, the introduced species, had a really high variance in its C13 or carbon values. So it indicated it had a really more variable diet, which we know about rats. They're very good at living in anthropogenic environments or environments that humans have affected. And this overlapped in some places this very wide dispersal of different species, or of different carbon isotopes, sorry, with a number of the Haitian forms. But what you can also see from these Haitian forms is that while there is some overlap, they do have a more, for the most part, a more constrained distribution, I guess I would say, of the carbon values here, indicating that they might have had a slightly tighter niche or a more specific diet. The one that we do also find with a really wide distribution of carbon values is this little Isolobodon species here. And it's a new species that hasn't really been described before, so we're not quite sure what to make of that. Yes?
So could some of the distribution be from just, so the one on the end is the rat, right?
Oops, sorry. Yep, there we go. Yeah.
Could some of the distribution just be from them being introduced to the island habitat like later than some of the other animals, so they're like newer maybe?
Potentially, and they're eating lots of different things because of that. Yeah. They also, the rats have been really, the genus Rattus has been really successful because they're omnivorous animals that are not really specialized. And the stable isotopes in these animals paint a picture of animals that might have been more specialized in their particular niche. And that specialization could have potentially occurred too because they're competing against each other for niche space. And to all survive and not go extinct, you have to have more or less non-overlapping niches.
Do you think that their niche will become more specific as time goes on?
The rats?
Mm-hmm.
Probably not. They're not competing against very many animals anymore because most of the stuff is extinct. Rats are, they're adapted really to be a generalist. Mm-hmm. So they can eat lots of different things. It's possible that they might become adapted to a more specific niche over kind of geologic time, many thousands or millions of years, but in terms of near time, probably not. Okay. So this small little new species that's outlined in blue here, and maybe there's a mixed assemblage, or maybe there's some juveniles, because animals that are still nursing have a slightly different isotopic signature. We're not quite sure. We need more fossil evidence to really sort that out. But this is a picture of that rodent species. Now, finally, just finishing up in the last couple of minutes with Caribbean extinction. So, I've used dentition and some postcranial analyses, which I didn't talk about in detail, to figure out how these animals were adapted to these specific niches. But today, almost all of them are extinct. And on Hispaniola, we just have the two surviving animals, Plagiodontia and Diem, which is a rodent, the Hutia, and then Selenodon, which is a shrew-like animal. And one of the major questions that some of my colleagues and I are interested in understanding is what caused these species to go extinct. And lots of researchers have been interested in this, and climate has been proposed, changes in climate, as a possible reason, as well as human activities. Things like clearing land, hunting, and the introduction of invasive animals like the rat. But there are also potentially genetic or anatomical features that might predispose a species to extinction. So things like body size. Certain animals of a certain body size might be more likely to be predated against, either by introduced animals or by humans. Or maybe there's other aspects of niche. If they're very specialized and the type of fruit tree that they get most of their diet from is goes extinct for some reason, then they would also succumb. So there's these different factors that might lead to extinction. And helping us understand these different factors, it will help us preserve the remaining animals. So better understanding extinction in the past can help us get a handle on what extinction looks like today. This is a reconstruction, again, by David Reaney, who illustrated here our primate, a little extinct shrew-like animal called Nesophontes, a giant ground sloth here, and then a rodent. I've been calling it Hexalobodon, which is one of the species in Hispaniola, but they probably all kind of look very similar to each other. And then this is a bat here hiding out in the tree hollow. So today, from Hispaniola, the primates are gone, the sloths are gone. There's one species of a shrew left, one species of a rodent, and then the bats have done better, 18 species of bats, so about 50% of bats have gone extinct, but 50% have survived. And my colleagues and I, this a couple years ago now, did an analysis looking at a lot of different factors, things like body size, in these animals to see if we could try to isolate some factors that would have resulted in their extinction. I'm just going to present the data about climate and human occupation of the islands, because we don't have quite enough data yet to say what factors that are intrinsic to the animal would have predisposed them to extinction. We're still working on all these data, so hopefully in a few years we'll be able to say something. But this figure comes from an analysis where we looked at the last appearance dates of different fauna based on radiocarbon dates of fossils, and the first appearance of human beings on the island based on radiocarbon dates of archaeological remains. And there are a few patterns that emerge. For fauna last appearance dates, the large mammals, so things like the sloths and the primates, for the most part, disappear around the same time that we see the first humans come to the islands. So these ones with the red stars are our sloths, and so we have Parachnus disappearing almost as soon as humans arrived on Cuba, and then Megalochnus just a little bit after. On Hispaniola, there's a similar pattern with some of the sloths. We can see them appearing. Here's the human bar and disappearing a little bit after. These two are kind of interesting. There are not very many dates for these species. So the dates that we have for Neochnus and Acrotochnus come from just one carbon date. So those, it's a little bit harder to say exactly when their last appearance date was, but they disappear a little bit before human arrival. I suspect with more dense sampling, we might see this move down a little bit, but we'll have to see. And then the primates, again, here on Hispaniola, they disappear just about 1,000 years after humans arrived, and almost simultaneously on Jamaica. There are a few outliers for animals that seem to have disappeared quite early on. We have some rodents here and some bats. The bats are an interesting case because some bats are adapted to very specific cave environments. And as climate changed and sea levels rose, some bats may have lost their habitat because those caves became inundated with water, and so that the hot, damp caves might have been less available for them. So we do see in Cuba a number of bats disappearing prior to the arrival of human beings, and similarly on Hispaniola. And then in Jamaica, there's this one outlier here, this guy, which is a very, very large rodent that disappears fairly early on. And we don't know exactly what if it's a sampling issue or there was some other factor that caused its extinction. But we do see a pattern of these big things disappearing, clustering around human arrival. And then there's a second wave of extinction of little things. So these are mostly bats and small-bodied rodents. And they disappear between 1500 and the present. And that is largely, we hypothesize, a result of the introduction of invasive predators and the destruction of habitat by European populations. So they introduced cats, dogs, mongooses as well, and these different carnivores would have preyed on these small-bodied rodents in particular. And rats may have played a role in forcing them out of different ecological niches. However, we're still collecting data on that. So again, if our project gets funded in a few years, I might be able to say more. Yes?
We have one question from Molly that says, some bats are migratory. How does migration fit into your work?
That's a really great question. I would have to ask my colleague Liliana Davalos, who is a bat specialist. So if you send a follow-up email, I can ask her and then get back to you. I believe that most of these bats, this is just my off-the-cuff answer, and it might be wrong. I believe that most of these bats do not migrate because they're in a more tropical environment. So I think that the migratory bats that we find in North America are not present in some of these islands, and so most of these guys are hanging around all the time. But again, I'd have to ask. All right, so that was my last slide. I'll finish up with my acknowledgements here. So there's lots of people. My work is very collaborative, and so I work with lots of different people who are specialists in isotopes and bats and cave diving and all of these different kind of things. So these are many of my colleagues who have contributed to that, and then we've gotten money over the years from a lot of different places. So these are all our funders. Thank you very much for tuning into my talk, and I'm happy to take any additional questions.
Yeah, thank you so much for spending your morning with us. I do have one more question from myself. So when you're picking your field sites, do you pick based on like kind of the continental movements that have happened in the past, or do you pick based on where other species have been found?
For field sites in the Dominican Republic, we basically pick places where there are fossils. So thinking about on a micro level, just on the island, there are some areas that have a karstic landscape, and karst forms caves well, and so you want to concentrate on those regions to look for fossils and look for potential regions where there's fossils. Now, picking on a kind of macro level, like why, if you mean like why did I pick field sites here in the Dominican Republic and on Hispaniola more generally, I was really interested in, I thought there were a number of interesting questions that were going on in the Caribbean. The questions of extinction, and it serving as a case study for extinction in near time was interesting to me. There was lots of biogeographically interesting things happening in terms of vicariance and dispersal and different kinds of land barriers or water barriers that were set up between the islands. So we could use the islands kind of as a case study for understanding evolution and adaptation of organisms to a local environment. And so for those reasons, I picked these sites to work at.
Great. And do you have any last things to add before we let you go?
No, I don't think so. This was fun. I enjoyed it. Anybody who has any follow-up questions, you can feel free to shoot me an email, and I'm happy to answer it. And I think that's about it.
Awesome. Well, thank you so much, Dr. Cooke, for spending your morning with us. That was a really fabulous talk, and I will talk to you soon.
Okay, I'll talk to you soon. Thank you so much for inviting me.
Yeah. Have a good afternoon.
You too. Bye.